Hello, everybody. This is a poster talk for a few minutes discussing SARS-CoV-2 in view of the in situ MS proteomics and integrative modeling. Let's get going. So this work was done with my colleague and friends, Dina Schneidman and Nir Kalisman, and I'll very briefly discuss what is this proteome that we know of today from SARS-CoV-2. Then I'll introduce the in situ CLMS protocol that we developed, and I'll illustrate how this can help us understand function and structure of three coronavirus proteins. So as of now, we know that there is a huge network of interaction once viruses are introduced into cell, as illustrated here. And this is just a result of two seminal papers from last year and the last year from mass spectrometry analysis of affinity purified proteins. However, there are issues that are unsolved, which are questioning whether the hundreds of interactions that you see here are indeed physiologically relevant, or whether those are direct or indirect interactors, as is illustrated here. And what we wanted to address is basically the question, which is what happened in real time, in real living cells, and can we gain structural information out of it? So to do this, we developed a method called targeted in situ CLMS protocol, and the idea is as follows. You start with a plasmid with a tag, then you transfect cell, and the important part is that you put an in situ cross-linking that will cross protein within the cells. Those are crossing the membrane. Once you finish this, you wash all the excess, and then and only then you lyse the cell, affinity purified, and go to the mass spectrometry. Of course, what you are seeking here is a combination of proteins that were cross-linked within the cell. The next phase is quite straightforward, in which we are using modeling, whether in a classical template-based docking, if we have one, or using this cross-link that we just recovered to do docking and structural studies. So this time I'll discuss the three protein, NSP1, 2, and N protein that belongs to this very big replicating proteins, and to a structural protein, which is the N protein. Now, the NSP1 is known to alt translation, and indeed we were able to show that using these pairs of cross-linked, we were able to show this structure here that I'll show you on the NSP1. Moreover, we found that it's bound nicely by this line, as you can see here, to the ribosomal S3. Therefore, we were able to get a detailed 3D refined model of NSP where it's bound to the ribosome and prevent the mRNA entry. The second important protein is the N protein, that its structure was known. However, we know quite a lot about it without really understanding how it contributes to this condensate formation that allows liquid-liquid separation and to build this very nice packing of the virus. In order to do this, we repeated our protocol on N protein, and we got this many paired, which confirm that this protein works as a dimer. Actually, it works as a dimer in which this part is very flexible, and you can see by molecular dynamics that it's really very flexible, while this is very tight. We were able, using this very detailed structural studies, to show that it has a very strong interface, which is the docking site for the RNA, as you can see here, the RNA in gold. Now, the last point is the NSP protein, which is solved just recently by AlphaFold in the Cas14. And basically, we know nothing about it. It's a very flexible protein, as you can see here. And this flexibility prevent to understand who is sitting next to who. And this was exactly what we did. Using the same protocol, we see that the AlphaFold model cannot fit the many clashes and contradicting cross-linked, as you can see here in red. However, once you break it down and you build it from scratch, you get a satisfied in which 98% of the cross-link are sitting in the right place. And that means that we were able to really understand how it's behaved in the cell. Of course, we showed also that doing in vitro and in vivo, in situ, in the cells, provide very, very different result. And as you can see, even model that was just came out from cryo-EM showed that it doesn't fit the reality. So it's a flexible protein, but in cell it's much more compact than anticipated. We were able also to find that it's a zinc finger by having this detailed structure. And to summarize, I just want to say that what we learn is that we learn about 3 proteins, but more importantly, we have a cross-linking method that captures 3D structure within the cell into an atomic resolution. And I want to thank you very much, and I'll be happy to take a question